Well, I joined the University of Michigan as an assistant professor in 1996, coming from a postdoc at UCLA. And I had been broadly involved in interdepartmental neuroscience programs throughout my training, both USC, where I did my graduate work, and UCLA had robust interdepartmental neuroscience programs. And so I became extensively involved with the graduate program at Michigan, was involved in training graduate students in my laboratory from both psychology and neuroscience, and wanted to contribute and give back to the program. And I thought the leadership role would give me an opportunity to shape some things that were going on in the program at the time. And at the time I took the directorship, there was an emergence on campus of several very large neuroscience focused initiatives. Those included the creation of an undergraduate neuroscience concentration, which was partnered between biology and psychology. And I had an active role in that process, served on the steering committee that resulted in the formation of that undergraduate concentration, which, as I understand it, is very popular. There were several life sciences initiatives being championed at the time, and those were in some cases focused around neuroscience. And I took the lead along with John Janitas and Hyland Moises in teaching a course that was called Brain Learning and Memory that we offered in the new USB neuroscience labs. So that was also happening at the time. There was the expansion and the construction of the undergraduate sciences building, neuroscience space, office space, classroom space in that facility. And so all of these things were, putting neuroscience really at the forefront of the conversation at the University of Michigan. And I thought this was a really important time to have a leadership role in that. And I took over from Peter Hitchcock, I believe, at the time. And he had seen many of those changes through that transition. But it was a very important time, I think, for the program. And I think it saw incredible growth, and I was happy to be a part of that. I think I started as an associate director under Peter, and then I did that for a few years, and then took on the directorship role after that. I had a great time directing the program. It's a challenging job insofar as there are not faculty lines associated with the graduate program. There's a lot of work gathering resources from deans, department heads, and so forth in order to make the program work. We had a training grant at the time. I don't know if you still have the same T32. And so that was an incredible amount of work to maintain and renew. So I saw it through one renewal at least. But those challenges aside, and I would say we were very successful in supporting the program and supporting students. No, it was a great experience. Personally interfacing with upper administration and having that experience was important for my own professional development. Really having, the sort of in the trenches contact with students in all aspects of their, growth and well-being was a very valuable experience. Really took a lot of pride in recruiting students, bringing them to Michigan, telling them about all the great resources Michigan has, seeing them settle into their own research programs and dissertation research and making that grow. So it was a really great experience. I haven't pursued a similar role here at Texas A&M, although it's been in the back of my mind at times. I've had other administrative responsibilities here that have distracted me, but I sometimes think about coming back into the role because I really did enjoy it. As you know, this job cannot happen without the support of Valerie and Rachel. They're absolutely essential to the functioning of the of the program, and just great people. So that was another thing that made the job wonderful. I think we were around class sizes of, I want to say 10 students. Don't quote me on that. One of the challenges that I faced is that despite the success of renewing the T32, the early neuroscience training grant, they cut our slots from 7 to 4, I believe, at that time. So we didn't shrink the class size. It just meant that we had to look to other places for support. But it was that sort of pressure as an IDP, as an interdepartmental program, that was always of the director's concern, is trying to maintain a class size with roughly, I think at the time, 95 training faculty, and trying to maintain the number of students that we were admitting each year, was always a challenge. During my period of time at Michigan, I was at Michigan for 16 years. And as you said, I left in 2012, leaving the door as the director of the program. And so from my perspective, the program had grown tremendously in the period of time I was at Michigan. Now, where it has gone since I left, I can't say, and I haven't been too attuned to what's happening there. I can say that we routinely compete for graduate student recruits with Michigan, down here at Texas A&M, and we're often on the losing end of that competition. Yeah, no, that was the first leadership position I held, and as I mentioned, it was a really great professional development growth opportunity for me in many different dimensions. And I think you have to be skilled at tactful negotiations with deans, department heads about acquiring resources. You also have to be skilled at getting faculty to commit their time and effort to a program that doesn't cover any of their salary. And so I was always really grateful to a number of faculty members that went above and beyond, contributing time and effort to the program. Jeff Murphy in particular, I will always remember as having made such an important contribution to the neuroscience boot camp course. And just like with the staff that I mentioned, the program really can't function, and I assume this is still true, without a very robust commitment of time from faculty that are just concerned about the student training experience and want to really expand neuroscience research and training on campus. I didn't feel like there was an enormous competition between my attention to the program and my research program. I didn't at that moment in time have a whole lot of other service activity that I was committing to the department. I think I might have been involved in our departmental executive committee and maybe involved in local promotion and tenure processes. But when I look back at it, I actually had quite a bit of time to give to service relative to what I have now, and so it really didn't put me in conflict with my research efforts. And in fact, I had neuroscience students in my lab, so it was sort of a nice symbiosis there in terms of working with them administratively in the context of the program, but also seeing them serving on their committees, seeing them through their dissertation training as well. So, I enjoyed the experience. I'm in psychological and brain sciences here, also a member of the interdisciplinary neuroscience program, which we call TAMIN, the Texas A&M Institute for Neuroscience. Also actively involved. All of my graduate students are neuroscience graduate students. I have currently 6 neuroscience graduate students in my lab. The environment in that sense, or the structure of the program, is very similar to what it was at Michigan in terms of an interdepartmental program, faculty from the medical school, College of Engineering. We have a very robust veterinary school here that has a number of neuroscientists, College of Science, College of Liberal Arts, which are separate entities, although we are merging them in the fall to one large college, similar to what is the model at Michigan, but it's a jarring change given that it's been separate colleges for the history of the university. So we'll see how that all goes. This is the perennial question: how do we take the work we do at the bench and bring it to the bedside? And there's no doubt that we have an incredible toolkit growing every day. I'm active on social media and I see the tools appear almost on a daily basis. And so we are positioned to learn an incredible amount about neural circuits and cellular basis of neurological disease, psychiatric disease, and have this incredible toolbox to really probe the system. And the question is, how do we now bring that to humans? And I think that there is an emergence of really exciting translational approaches to do that. And certainly some of the chemo and optogenetic approaches that have been pioneered in the last decade are amenable to transport into humans. And there's work that's begun in some of those domains and would potentially achieve much more circuit specificity than we now have with standard pharmacological approaches for psychiatric disease, for example. So, I mean, I really think we're on a cusp of a revolution in terms of how we can now transform, the therapeutic interventions for diseases which we've not had much progress on, in the last 20 years. It's a challenge though in terms of training. So as the neuroscience students coming into the discipline are confronted with such an array of methodologies that are changing frequently. And so it really does present a major training challenge in how you maintain, how you develop expertise in these methods and what students choose to pursue and what they choose to focus on. And all together, I think they need much more computational and quantitative experience across the board. And so how you bring that into play, because everything is generating big data now, whether you're doing mini scope recording, imaging hundreds, if not thousands of cells, or human fMRI, large data sets, or transcriptomic analyses. It needs people who can crunch numbers. And so I think that's been a major challenge for neuroscience training programs, is how do we get students actually coding and dealing with large data sets, because I think they need those skills. But back to your original point, I think we are at a moment where we can start really using these tools that have accelerated progress in understanding brain function in animal models and bringing that into translationally relevant domains. The most memorable experiences for me were without a doubt the annual retreats. I mean, so the annual retreats, which in my time happened at Walden Woods in Howell, Michigan, or at the Kellogg Biological Station, kind of out by Kalamazoo. Those were without doubt the highlight of the experience. So really peeling away from campus, heading out for an overnighter with the students, the staff, other faculty. Yes, we did a lot of science and we had posters and talks and that kind of stuff, but we also had a lot of downtime. And one of the most exciting events for me associated with the retreat was the tug of war. And the tug of war was always an opportunity. I can't remember if we pitted students against faculty or if we were intermixed or how it worked, but that was always a highlight. And so that tradition, I hope, continues. It was a great tradition and a whole lot of fun. And I think at the end of the day, if you're not having fun, you're not doing it right. So we need to be able to enjoy these experiences. Graduate years are outstanding years of one's career. And, I really like the retreats and the opportunity to share some of that with the students.